Full screen.
All right. There we go.
There.
Okay. So Orion seemed to be intrigued by nature at an early age. Whether he was going to go into the field of studying insects or perhaps crustaceans, as his shirt today might indicate, that could have been an alternative path. He was very interested in the water and the ocean, and he and his sister enjoyed playing with marine critters in their bathtub, so maybe he would go on to being an experimental biologist. He mentioned that he and his sister, I think maybe when they were 7 or 8, started trying to sell their art so that they could use the money to help save nature. So he had an early conservation spirit. But there was a point in time where maybe he would go on to be a dolphin trainer at SeaWorld. Luckily, he did not take that path, luckily for us. But he always enjoyed spending time at the water and seemed to love the beach. I think he said the first time he went snorkeling, he fell in love with the ocean, and that may have been what helped to seal the deal in terms of deciding his career path. And so he became very excited about snorkeling and swimming. You see him donning his little blue fins, which will be something we will see continuously throughout Orion's career. I think he's owned those same blue fins his entire life, and he wears them every time we go diving. So he fell in love with the ocean. The first time that he got to experience a coral reef was during a study abroad program in Australia. And that's where he really fell in love with coral reefs. And so off we go to Orion's career. He started, and there's the little bluefin. Those are the ones we see every time we go in the field. So he did his undergrad at the University of Maryland in biology and ecology and evolution. He had a study abroad at JCU in Australia. He worked for a bit at Meridian and WWF, kind of beginning his interest in merging coral reefs with conservation science. Back in 2018, I believe, Ryan was applying to grad school, applied to a number of different programs. He was awarded a National Science Foundation Graduate Research Fellowship, which is a very prestigious award, and we were lucky enough to recruit him to come here to Scripps. So he joined my lab back then, 2018. Our lab was relatively small back then. It's not small anymore. Ryan's also had the fun. Well, he wasn't there, so this was this year. He's also had the pleasure of joining not just my lab, but the collective Smith and Sandon lab family. This was back in before COVID, I think 2018 or 19. This lab family is not that small anymore either. So we've really enjoyed having Orion be part of the family. Since Orion arrived at Scripps, or he moved from DC, where he came over to San Diego, spent a lot of time in Hawaii, Palmyra, even went to Egypt and to Micronesia. So he's had quite a round the world tour during his graduate career. But mostly for his thesis, he took over a large long-term monitoring program that I began on the island of Maui and associated neighbor islands, where we spent a lot of time in the field, underwater, collecting data, organizing teams of people, and just Orion really embraced this project and ended up taking over the lead of that work. So as I said, Orion spent a lot of time underwater collecting data, although I'm not exactly sure how he did that day. But I can promise you he wasn't always going around flying in the water. But you can also see pretty much everywhere you go, every picture I could find of Orion, he's pretty much smiling. So it seems that he really embraced this work. He enjoyed the field work. He enjoyed training and mentoring others. Again, the smiles just seem to be contagious in this team. So you might wonder, or I wondered, maybe it's because of all those tropical drinks that our island partners, such as Donna Brown here, is making us. We have a lot of tropical drinks at Donna's house, or maybe those Mai Tais from the the Tommy Bahama bar, which is unfortunately no longer with us, from Lahaina. But no, it turns out this is just a theme in Orion's life. He's always smiling with friends, family, significant others, office mates, neighbors. Again, everywhere you see, even sitting on Santa's lap, Orion seems to be happy. And even when Orion is actually Santa himself, he is also still happy. Orion also passed the prerequisite of joining my lab, which is being a dog lover. You can see many dog friends everywhere, including in here today. So that was great, and obviously smiling with those critters. Orion also learned the appreciation of malasadas, which if you haven't had one, if you did have one, you would have that same expression on your face. They're just incredibly delicious treats, sweet treats. He also gained an appreciation for top predators, and somehow onions. I'm not really sure what's going on in this picture, but I do want to hear that story sometime. I couldn't avoid putting that picture in here. But like I said, Orion had the pleasure of going to Egypt to join the UCSD Scripps COP delegation this past year, 2022, which was an amazing experience. And he also had the pleasure of joining us on a field trip to Palmyra Atoll, which of course, lots of smiles there, a really unique place. All of this work does eventually make Orion tired. I was able to find a few pictures of Orion sleeping, maybe relaxing a little bit too much. But even when he's sleeping, he's still smiling. It's funny. I don't know really what's going on. So all that being said, Orion spent an incredible amount of time underwater. He's gained so many skills since he's been here at Scripps. He's been a pleasure to have in the lab. He's been able to work in both tropical and temperate water environments with those little blue fins. I guess they didn't make it into cold water. Yeah. And he's also been able to hone the skill of like never aging. Every single picture, he looks the same as he did when he was a kid. It's amazing. But no, it's been a real pleasure of having Orion join the lab. He has been incredibly productive, publishing papers, getting awards, mentoring, I don't know, dozens of students, and just being a great family member. So thank you, Orion. And with that, I'm happy to pass the torch and let you tell us your story. You want me to hide this?
Okay. Thank you. That was quite the introduction. It's not expecting to see so many young pictures of me. Let's see. Can everyone on Zoom hear and see the slides? Just want to make sure before I launch into it.
Yes.
Yes.
Yep.
People on Zoom hear us?
Yes.
Awesome.
Yep.
Okay. Let me. Where's the 1 second? Need the presenter view. Let's see. 1 second. Oh. It should work. I can change the size of that.
I know, but I think if you just
1 second, bear with us.
sharing.
You just do screen 2, does that work? Thank you. Thank you. And I'm still.
Why don't you try closing it and starting?
Oh, maybe it's both of them up at the same time. Okay. There we go. Okay.
And make sure that, are we sharing it in Zoom?
One more time, can people in Zoom see you
got
can
it.
see there? Great. Okay, with that anticipation built up. Thank you guys. Thank you, Jen, for that introduction. And I want to thank everyone for being here today, both in person and on Zoom. It's really means a lot to see everybody here, especially after 5.5 years working on this. So I'm really excited to talk about the research that I've been doing and to share updates on the resilience of coral reefs that I've been studying in Maui and the Hawaiian region more broadly. So I'm going to start with this broad overview of the research that I've been doing since joining Jen's lab at Scripps. And the dissertation that I have worked on consists of 5 interrelated chapters, which range from classic ecology topics such as scale, succession, and selection to applied social science focused on reducing the barriers to using conservation technology, to applied conservation science that's focused on evaluating the efficacy of a conservation planning tool. And I won't be covering all of these chapters today in detail, particularly the ones that are already published. Instead, I'm going to focus on telling a bit of a story about how multi-scale ecological data and conservation technology can be used to inform coral reef conservation. And by the end of the talk, my hope is that the data I've shown you can convince you that there's hope for coral reefs and a need for evidence-based conservation. So Jen already covered a lot of this, but the basic message I want to get across is that I've been in love with the environment for a really long time. And through undergrad and some pivotal internships and work experiences, that love transformed into an interest in applied conservation and the science of how we can connect ecology and conservation together. And it was my time in Australia and a coral-focused internship after graduating that focused my interests on coral reefs and marine science specifically. And I mean, given how ecologically incredible coral reefs are, it's hard not to be captivated by them. And just as one really cool fact, 1 in 4 marine species spend some portion of their life cycle on a coral reef. So these are just really incredible ecosystems. And from a human perspective, they're really important as well. They support fisheries, tourism, and coastal protection benefits for more than 1 billion people around the globe, and they have really immense cultural value for coastal communities. Unfortunately, reefs do face a wide range of different threats, and this includes threats like overfishing and nutrient pollution that are tied to local human impacts, which are shown here in purple, as well as global scale threats linked to climate change that are shown here in yellow. And importantly, these threats can interact with each other, and a reef may be exposed to multiple different stressors at a given point in time. And these stressors have already caused major degradation on coral reefs worldwide. It's estimated that about half of all coral cover has been lost since 1950. And as climate change worsens, nearshore human impacts increase, reefs of the future will face threats that are more severe and synergistic with one another. So given this, all this information, it's no surprise that I'm often asked this question: are coral reefs doomed? Why study coral reefs or dedicate resources to conserving them if coral reef decline is inevitable? And I'm here to tell you that coral reef decline is not inevitable. And while coral reefs are indeed threatened, that doom and gloom narrative misses 2 key points. First, coral reefs are dynamic ecosystems that change over time in response to disturbance. And second, no 2 coral reefs are the same. Different regions of the world and even different islands, or same coastline within the same island, have different coral reefs that have different species composition, different histories of environmental disturbance, and unique human impacts. And multiple studies have identified specific reefs or regions with signs of resilience, and conservation that can focus on identifying those resilient reefs and prioritizing them is one of the key focal points for reef conservation going forward. Now, I want to take a moment to specify what I mean when I say resilient, because I know that can be a bit of a squishy term. And as applied to coral reefs, resilience is typically defined as a reef's capacity to resist stress and to recover from stress. And if a reef was a person, this would basically be, resistance would be their ability to avoid getting sick, and recovery would be their ability to get better after falling ill. And this resilience is determined by multiple different ecological processes. And if we measure the right processes across multiple different reefs, we can predict which reefs will be the most resilient. And then we can concentrate our efforts to reduce local human impacts at those reefs that have natural resilience so that they can better withstand climate change in the future. And this approach is termed resilience-based management and has been widely used to target coral reefs for conservation, including by my former colleagues at the World Wildlife Fund. And now resilience-based management is a form of conservation triage. And conservation is almost always resource limited, so it's often necessary to prioritize specific locations or species for limited conservation resources. Listing a species as endangered or putting a location within a protected area are also examples of conservation triage. And for triage to be successful, we need to have confidence that we're choosing the right species or locations to protect. And so monitoring data can be used to evaluate these decisions and improve future ones, but this all depends on having access to quality data. So to illustrate this point, I'm going to ask you all to participate in a little exercise that has nothing to do with coral reefs. We're going to bet on some good old-fashioned horse racing. No money's going to change hands. I just want people, if they're on Zoom, to use the chat to write which horse they think they're going to win. If you're in person, you can put the number up with your fingers. If you think it's a double-digit number, use your dive training to figure out how to do that with your fingers. And you can see the numbers of the horses down there on the bottom. So. Okay, we can get ready. Put your fingers up. Put your hands up. Okay. Okay, and they're off. If you haven't written your guess or put your fingers up, it's not too late. Number 2 is ahead right now, but neck and neck with number 13. Look at him go. Number 7 just advanced to the lead, but number 1's closing in fast. Look at that stamina. They're coming up to the finish line, and it's almost over. There we go. Number 1 has cinched it. Okay, so who got it right? Show of hands. Be honest. Nice. Okay. Okay, so not very many people got it right, even though some people have watched this presentation before. But as spectators of this race, I didn't give you much information to go off of. So it's not that surprising that we were all over the board. If you had unlimited money, you could have spread your guesses across multiple horses to ensure that you still bet some amount of money on the winning horse. But I only gave you one guess. So if you wanted to really increase your chances of guessing correctly, you'd want to base your decision on more information than just a horse's number. But does more information always give us a better chance of picking the winning horse? Not necessarily. If we had information that wasn't functionally related to a horse's performance, our predictions wouldn't be any better than just randomly guessing. For example, knowing a jockey's favorite color is not going to help us pick the winning horse. Now, instead, let's say we have information that is indicative of a horse's performance, but that information has a lot of intrinsic variability. For instance, if we measured a horse's heart rate, but just one time a month ago, that single value on its own won't tell us a whole lot about which horse is going to win because it can vary so much over time. Similarly, any predictions that we make based off that one value won't be very precise. To really make an accurate and precise prediction, we need metrics that are functionally related to a horse's performance, and that information needs to be collected at the correct scale. And that same concept is true for coral reefs. To accurately predict whether a reef will be able to resist and recover from stress, we need indicators of reef function that are measured at the correct scale. And so one example I'm going to talk about here is called habitat structural complexity. So corals grow to form these complex living structures on a reef that can provide habitat for other organisms. And I really like this picture because it shows how a single coral can provide habitat at multiple scales, both for the tiny little fish that live inside a coral's branches to the large fish that's living underneath the whole coral colony. And some of these fish, maybe not these specific fish, but some reef fish graze on algae, and that helps to control the balance between corals and algae on the reef. And so structural complexity is a functional indicator of resilience because it says something about habitat availability for other organisms, but also about the ability of the reef to support healthy herbivore populations. However, corals do not create structure across all scales. And this figure is from my first year project here at Scripps, where we basically measured the structural complexity of 60 different reefs across the Hawaiian archipelago at different scales. And here you can see that at the scale of just a few centimeters, which may correspond to the small branches or interstitial space of a coral colony where a crab or baby fish might hide, we've got a strong relationship between coral cover and structural complexity. So the one all the way on the left, where an increase in coral cover is an increase in structural complexity. Then as we go to slightly coarser scales, we see this relationship change a bit, where there's still a relationship, but that peak in structural complexity is at intermediate values of coral cover. And then when we get to even coarser scales, that relationship between coral cover and structural complexity disappears entirely. And instead, we see a relationship between the overall topography of the reef and structural complexity. So suffice it to say, if we're interested in coral-driven habitat structure and the functional relationship that has with resilience, we need to make sure we measure structural complexity at the appropriate scale. Otherwise, our metrics won't correspond to coral-driven structure at all. If you want to learn more about this project, I encourage you to check out our paper in Marine Ecology Progress Series, but I do think this project does a good job of illustrating why I was interested in coming to join Jen's lab at Scripps. Jen brings a rigorous ecological perspective to all of her research, and her lab is broadly focused on solutions-based marine science. And furthermore, both our lab and our close collaborators in the Sandin lab use 3D modeling technology to study nearshore ecosystems across multiple scales in space and time. And our labs maintain multiple long-term monitoring programs using this data, including the monitoring program in Maui that Jen mentioned that I've been managing as part of my PhD. And these data provide exciting new ways to study how reef processes are functionally related to resilience. So a quick overview of how this technology works. When we go into the field, we swim above the reef with a camera rig. We take a picture of the reef every second and aim to get good overlap, swimming over an area of reef about the size of this room. And here you can see the diver swimming back and forth. We don't swim quite that fast, but we do aim to get comprehensive coverage of the reef as illustrated by these pictures here. And you can see how a single feature on the reef, as shown with this red circle, is captured from multiple angles in successive pictures. And then when we get back to the lab, we use an algorithm to stitch the pictures together based on that overlap to create an accurate 3D model of the reef floor, reconstructing only things that are stationary and not moving. Now, from that model, we can create other data products, such as a digital elevation model or a big 2D map of the site. And collectively, this group of 2D and 3D products fall under the umbrella of large area imagery. And I'm going to keep using that term to refer to this 3D modeling technology going forward. And it's really great for studying processes across scales, both in space and through time. So if you revisit the same reef year after year, collect these images over the same area, you can put them together into a visual like this. Whereas we move from left to right, you can basically, you're moving forward into the future. You can see the impacts of multiple bleaching events here where corals turn white during spikes in ocean temperature. And you can see how some corals recover after bleaching, others die, and others don't even bleach at all, even though they're facing pretty similar environmental conditions. We can also use this technology to look at the impact of a disturbance event on the entire reef. This is a different reef from Maui that was damaged by a historic swell event in 2022. And here, red indicates parts of the reef that were damaged and removed due to that wave event, while blue indicates new growth. And visualizations like this allow us to better understand and communicate the scope of reef damage or growth. Here you can see an entire coral colony, pretty large, that was just entirely removed by waves, leaving just a crater in the reef. So to give you a sense of how this technology has been applied to study coral reefs to date, I want to share this figure from a literature review that I led as part of chapter 1 of my thesis. Here in the middle, you can see a variety of different topic areas related to coral reefs and how often they've been studied using this technology. And you can see that testing the accuracy of this technology and studying habitat structural complexity are two of the main ways it's been applied. And this isn't terribly surprising. This is a 3D model after all. It lends itself to studying habitat 3D structure. But there's a bunch of other topic areas with conservation relevance that are currently understudied, and I'm highlighting conservation efficacy here in green because it's a focal point of this talk. Importantly, to complement this literature review, we conducted a survey of conservation practitioners and asked them how they would like to use large area imagery. And we found quite a disconnect between their interests and the current applications of this technology in the literature. Through this survey, we also got a sense of the barriers that some of these folks face to using this technology, including the cost of equipment, technical capacity, and cost of data storage. And so there's both a need for applied conservation, but also efforts to increase the equity and access of this technology so that we all can have access to this transformative tool for conservation. So as part of this project, we developed some specific recommendations that the scientific community can take to improve access and increase the conservation uses of this technology. And if you're interested in any of that, I encourage you to check out our paper in conservation biology. So we've established that large area imagery is an important tool for measuring reef function across scales, and that it should be applied for more direct conservation applications. We've also established that forms of conservation triage, such as resilience-based management, need to be evaluated so that we can ensure the right locations are being prioritized for protection. All that we need to pull this together is an existing large area imagery time series that overlaps with a resilience assessment that's already been conducted. Luckily for my thesis, we had just this scenario on the island of Maui. In 2018, just as I was starting my PhD here at Scripps, the Nature Conservancy and the Hawaii Division of Aquatic Resources conducted a resilience assessment on the west coast of Maui, which you can see their sites here. Their team surveyed more than 24 sites using traditional transects and photoquadrats, and they measured 7 ecological metrics, which I'll go over in a second, and combined them into a single predicted metric of resilience, and then rated sites from high to low resilience, as you can see here. Now, 6 of their sites happen to be located very close or directly overlap with some of our lab's long-term monitoring sites that use large area imagery, and those are shown here with the white arrows. So I've been working with the Nature Conservancy, Division of Aquatic Resources, and the UC San Diego School of Global Policy and Strategy to validate the predictions of this 2018 resilience assessment. So basically, we wanted to find out whether those predictions are accurate and precise, given that that assessment relied on a single snapshot of coral reef condition. Thinking back to that horse racing example, do we have enough information to predict the winning horse based on a single snapshot? Are we capturing information about the right functional indicators at the correct scales? So let's briefly talk about those indicators and how they relate to reef resilience. The first indicator I want to mention is the percent of the reef that's covered with live corals. And that's one of the most common indicators of reef health used in coral reef ecology and conservation. And since corals form the structure of the reef, higher coverage of living corals is generally assumed to be indicative of a more resilient reef. Similarly, a more diverse reef has species that fill the same niche, which is known as functional redundancy. And on a diverse reef, if one species disappears, it's more likely that another species will be able to fill in that gap. In addition to corals, the reef is composed of calcifying algae called CCA that helps to form the structure of the reef and bind it together. And the balance between these calcifying organisms and the fleshy algae reflects the state of competition on the reef and whether or not it's actively growing. The presence of baby corals is also an indicator that coral populations are actively growing, while coral disease could indicate that corals are stressed or unhealthy. And then our final 2 metrics are herbivore fish biomass and rugosity. And rugosity is just a way of quantifying habitat structural complexity. And I already described how these 2 metrics are thought to be functionally related to resilience earlier, so I'm not going to go into detail on those here. So basically, the Nature Conservancy and Division of Aquatic Resources measured these 7 metrics during in-water scuba surveys. But for this project, for us, we were able to quantify the first 6 of them directly from our 3D models using 5 different time points. Unfortunately, we didn't have enough data to quantify herbivore fish biomass since we didn't have separate fish surveys for all of these sites in those years, and because fish are not reconstructed in the 3D model because they move. So for this study, we used the first 6 indicators and we recalculated the Nature Conservancy scores using their values for those first 6 indicators as well. To do that, we basically normalized each of these indicators with respect to the highest value of each site so that it was the max value would be 1, and then we did an unweighted average for each site of all those indicators to get a metric of resilience, so each site had its relative resilience with respect to the others. Now, before getting into the results of the resilience assessment, I want to take a step back and provide some context about our large area imagery time series and the Maui Nui region in general, so the results of the resilience assessment make a bit more sense. The white arrows here, again, they show those focal sites where our sites overlap with the Nature Conservancy, but our monitoring program is broader and spans the leeward coasts of the neighboring islands. So we have Maui, but also Molokai, Lanai, and Kaho'olawe. And these islands have a shared history with each other. As recently as 10,000 years ago, they were all connected to form a single island called Maui Nui. And today, these islands shelter each other from open ocean swell, and that allows these really spectacular high coral reefs to form in that protected basin where our sites are. And it isn't uncommon to find reefs with 70 to 80% coral cover here, which is quite a bit higher than elsewhere in Hawaii or the broader Pacific. This region's also a really interesting study or case study of human impact. You have Maui, which has a lot more people than the neighboring islands, about 20 times more than Molokai and 50 times more than Lanai. So you have reefs on Maui that are more exposed to overfishing, nutrient pollution, coastal development, and threats like that. And as a result, coral cover on Maui's reefs tend to be lower than the neighboring islands. Kaho'olawe is a bit of a special case. It is currently uninhabited, but has historically been highly impacted by the US military, and they used to actually bomb that island, and by the grazing of invasive goats and other ungulates. So active restoration on Kaho'olawe is actually ongoing, and it's protected currently within a fishing exclusion zone. So back to our monitoring program, we've been using large area imagery to monitor these sites. For Maui, it's been annually since 2014, and for the neighboring islands, it's been every other year since 2017. And here I want to give a huge shout out to Jen's first PhD student, Dr. Emily Kelly, for starting this monitoring program a decade ago and having that foresight. All of this research would not be possible without that. And I also want to thank our lab's Maui family for helping us complete our fieldwork each year. It's been truly a highlight of my time at Scripps to get to know these sites and these people over the course of many years. And this close connection made it that much harder to accept the headlines on August 8th of last year, when wildfires ravaged Lahaina and claimed the lives of 100 people. Lahaina was our home base for fieldwork each year, and seeing videos of it go up in flames were truly devastating. And while none of our partners lost their lives, many lost property or their livelihoods. And this fire serves as a stark reminder that climate change will not only test the resilience of our ecological communities, but of our human communities as well. And furthermore, toxic ash, runoff, and debris from the wildfires currently pose unknown risks to human health and the environment, including nearby coral reefs, highlighting the need for data-driven conservation in Maui. Now, the fire occurred 1 month after our most recent surveys in Maui, so none of the data I'm going to share today reflect impacts from the wildfires. We do have one site here at Wahakuli that is fairly close to the burn scars. So on our next return visit, we might expect to see more impacts at that site compared to other sites, but we can't be sure quite yet. A few other site-specific circumstances to be aware of. Our sites at Kahekili and Keawakapu are close to some large coastal developments. And in particular, our site at Kahekili is just offshore of a wastewater treatment plant and has suffered from elevated nutrients and occasional algae blooms. And so to counteract that, the state actually imposed a herbivore fishing restriction in that area in 2009. So fish populations there are a bit different than elsewhere. And then down at this reef at Molokini, that's also within a protected area where no fishing is allowed. And because that reef is a bit offshore inside a partially submerged crater, it's actually exposed to less nearshore human impacts, but it does receive quite a bit of human traffic from dive boats and snorkel tours. So with all of this context in hand, I want to turn our attention back to our resilience assessment. And these numbers here show the relative resilience that was predicted for each of our focal sites. And this is based on that Nature Conservancy assessment from 2018. Now, 1 denotes the site that's predicted to be the most resilient. Now, let's compare those results to the 5 different resilience assessments we were able to conduct using different years of imagery for our time series. So each of these graphs corresponds to a different site. You can see the dates down at the bottom, and you can see how the relative resilience of different sites fluctuates over time with respect to the other sites. And while there's some amount of wiggle, the best and worst performing sites remain pretty consistent over time, and consistent between the large area imaging methods and the Nature Conservancy's predictions. For example, Molokini and Keawakapu over there on South Maui are consistently predicted to be more resilient, whereas you have Wahakuli and Olowalu that tend to show lower predicted resilience. And this consistency in our predictions is encouraging. It suggests that those functional indicators we're using for the resilience assessment are fairly stable over time and not highly variable like a horse's heart rate from our example before. But are these predictions actually tracking observed change on Maui's reefs over time? To answer that question, we need to be able to quantify resilience so that we have something to compare our predictions to. And for that, we chose to break resilience out into those 2 components of its definition, resistance and recovery. And so I'm going to use this red and blue color coding for resistance and recovery going forward. Red doesn't mean bad, it just means that it's relating to resistance. Now, next we have to determine what disturbance we're quantifying resistance and recovery with respect to. Our time series has captured a decade of major events on Maui's reefs, including 2 marine heat waves that caused coral bleaching, at least 2 large wave events, and the impacts of COVID-19 lockdowns and how those changed human behavior on the island. As a reminder, our data was collected before the fires, so none of those impacts are reflected here. Now, to check our resilience assessment predictions, we're going to quantify resistance and recovery with respect to that first bleaching event in 2015. And that's because this was the largest single disturbance to impact Maui's reefs over the past decade. And because it was the first disturbance of our time series, it gives us the longest time period over which to observe recovery dynamics. Now, a quick aside about coral bleaching. Bleaching can occur whenever corals get stressed, but it most often is seen in response to elevated ocean temperatures. And when this happens, corals lose their partnership between symbiotic algae that live inside their tissues. And these algae under normal conditions are a coral's primary source of energy and nutrition. But bleached corals, because they don't have these algae, will eventually starve to death unless water temperatures return to normal. And it's important to note that a bleached coral in and of itself is not dead. It has the capacity to recover if the water temperature goes back down, and it's been able to survive until that point, and then it can regain its symbionts. So turning back again to our multi-scale data, we can harness the multi-scale power of our large area imagery time series by tracking resistance and recovery at multiple scales, both the scale of individual coral colonies and the entire benthic community. And benthic community basically just means all of the organisms that are living and growing on the seafloor. So let's focus on this community scale first. To quantify resistance, we can measure what species make up that benthic community before and after bleaching, and then see how similar those two communities are to each other. And from a coral-centric perspective, we can also measure how much coral cover declines immediately after the bleaching event. To quantify recovery, we can measure how community composition or coral cover has changed in the 8 years since the bleaching event, and to see whether or not the community is returning to its pre-bleaching state or whether it's continuing to diverge. So I want to show you an example of some actual data for this focused on coral cover. Here you can see each of our 6 sites, and the y-axis is showing the relative coral cover for each site, where 100 is showing coral, that's the amount of coral cover before the bleaching event. So if it goes down, it's down relative to that pre-bleaching state. And the red line here shows the magnitude of that initial decline. And you can see all the sites lost coral cover due to the bleaching event, but the magnitude of that decline was different. Kahekili had a very small decline, so that site has high resistance, whereas Wahakuli had a larger decline, so that site has lower resistance. Remember, resistance is how much that change how large that change was after the disturbance. So now the blue line here shows recovery, or how coral cover has changed in the 8 years since bleaching. And you can see how some sites exhibited strong recovery while others continue to decline. And you can also see how the magnitude of that initial decline is not necessarily related to the recovery trajectory afterwards, which suggests that resistance and recovery are being driven by separate processes. Furthermore, if we add in our resilience predictions from the 2018 assessment, we can see that they don't necessarily align well with either observed resistance or recovery. So I want to focus in particular on this site down here in the corner, Keavakapu, which was predicted to have high resilience, but had low resistance and low recovery. And at this site, over our time series, we've watched as benthic community composition has shifted, with a loss over time of these orange encrusting Montipora corals, which have generally been replaced with calcifying CCA algae. And this decline could be driven by a number of different factors, including different bleaching susceptibility of those local corals, to localized nutrient pollution, to even outbreaks of coral predators. To get a better sense of what's driving that decline, we can track the fate of individual coral colonies at that colony scale. So for resistance, we can look at colony scale patterns of coral bleaching and whether or not corals survived. And for recovery, we can look at coral growth over time and see if certain sites have higher growth rates than others. Now, rather than consider the impact of the 2015 bleaching event in isolation, we can use our time series to look at the cumulative impact of multiple bleaching events on Maui's corals. So this is showing the bleaching response of 4 different key species of corals in 5 different years of our time series. And you can see that there was also bleaching in 2019, not just 2015. Now, these sites in Maui experienced very similar levels of heat stress between 2015 and 2019, but we see less bleaching in the second event, which could be a sign of acclimatization or selection. So if we can identify which sites show the most signs of acclimatization and selection, this could be a good proxy for resistance. So let's look at how individual corals fared in 2015 and 2019. And we can split this data across our 6 key sites. And we can see that here the different colored bars show different portions of the population with a very similar bleaching response over time. So for example, the dark green represents corals that we were tracking that didn't bleach in either 2015 or 2019, whereas the light green shows all corals that bleached in 2015 and then bleached less in 2019 or didn't bleach at all. And so those are the ones that could be acclimatizing or having some less severe bleaching response. Gray represents corals that we started tracking at the beginning of our time series and didn't survive until the end. And so I'll mention that heat stress was very consistent across our sites. So this amount of variability in bleaching response is notable and suggests that some sites have higher instances of acclimatization, thermal tolerance, and survivorship. And there's a lot of interesting findings relating to bleaching and acclimatization that I could dig into here, but for the purposes of this talk, I'm going to move on. But if you're interested in any of this, we submitted recently to PLOS One, and hopefully that will be out soon, so keep an eye out for that. What I do want to touch on is how this relates to resistance and recovery. So I've summarized the results from our PLOS One paper in terms of bleaching susceptibility, survivorship, and growth. And you can see green triangles are showing sites with significantly better rates in terms of those three metrics, and red is significantly worse. And you can see it's broken out by the 4 key species that we've been tracking, so we can see if certain species are driving trends at different sites. And some sites like Kahekili consistently performed well, while other sites like Wahakuli had low resistance but pretty good recovery. And these results largely track with our community scale data, and it helps us to determine what processes and species are driving resistance and recovery at these reefs. For example, if we look back at Keavakapu, that site I showed us the video of earlier, you can see that it looks like low survivorship and low growth is hampering that site rather than increased bleaching susceptibility. So let's bring this all together. Here's a map of our 2018 predictions from the Nature Conservancy. And remember, those were pretty similar to our predictions using large area imagery at different years. And remember, these are all based on single year snapshots. Now, if we compare that to the actual patterns that we observed of resistance and recovery over an entire decade, we can see a very big disconnect between predicted resilience and both resistance and recovery. So this is not a great sign if we're using this method as a form of conservation triage. So we're left wondering now, what went wrong? Why didn't our predictions line up with our observations? There's a few potential explanations here. One obvious one is that we're combining resistance and recovery into one metric of resilience, but those two processes are behaving very differently. And so if we were to model them separately, that could both increase the accuracy of our predictions and make them more informative. The poor performance of our resilience assessment could also be because we aren't using the right indicators, or maybe they aren't functional indicators. Did we accidentally settle on a crappy indicator like a jockey's favorite color that isn't very functionally linked to performance? It's possible, and it's also possible that we're not including some key indicators such as herbivore biomass, which we didn't have enough data to include. But I will say that the indicators we used have well-documented functional links in the literature, and they also have been used in a lot of other resilience assessments. So I want to propose this third option that perhaps we're using solid indicators, but faltering a little bit in our interpretation of them. Coral reef ecology is complicated, and metrics such as coral disease, structural complexity, and coral diversity aren't always one-way indicators of resilience in all circumstances. For example, coral disease has been shown to be an indicator of degradation and poor water quality, but it can also be spread when there's a high density of potential hosts, such as on reefs with high coral cover. Similarly, structurally complex corals, like delicate branching corals, may be more susceptible to wave damage than more robust corals with less structural complexity. And then also, chronic degradation can weed out more sensitive corals, creating a reef with less diversity, but more stability due to the dominance of stress-tolerant individuals. So to illustrate how this can play out, I'm going to zoom out from our 6 resilience assessment sites and look at broader regional trends using our entire Maui Nui time series. So on the left, you have our 36 sites from Maui Nui, and the coral cover at those sites is shown in 2017. And you can see some clear spatial patterning where the reefs at Molokai, Lanai, and northern Kaho'olawe have higher coral cover, and the reefs on Maui have lower coral cover. And if high coral cover was always associated with high resilience, we would expect to see coral decline on Maui and stability on the neighboring islands over the intervening period. But when we look at how coral cover has changed over the next 6 years, the results are far patchier with high and low performing island reefs on the same island. So initial coral cover appears to be a poor predictor of reef resilience, at least at this time scale. Now, if coral cover isn't providing enough information, we can dig a bit deeper and use species composition to make sense of how these reefs are changing. And for my chapter 3, we basically use species composition to identify 3 different types of reefs in Maui Nui. The first one had lower coral cover, about 30 to 40%, and was dominated by stress-tolerant Porites corals. The next reef type had more intermediate coral cover, about 50 to 60%, and had a lot more coral diversity and structural complexity. And the last reef had the highest coral cover, usually over 70%, and was dominated by these encrusting Montipora corals. So using our resilience indicators, community types 2 and 3 probably would have been predicted to be more resilient, and community type 1 probably would have been predicted to be less resilient because it has some hallmarks of degradation: low coral cover, low structural complexity, high turf algae, low diversity. We can use our map again to look at how these different community types are distributed around Maui Nui, and we can see some similar trends to before, where those low coral cover Porites reefs are more on Maui, the higher coral cover reefs are on Molokai and Lanai, but there is a bit more spatial variability here, a bit more patchiness. You can also use these box plots to look at how coral cover has changed in between each of our surveys. And this red line here indicates no change in coral cover. If you're above that line, it means coral cover increased between our visits. Below means it decreased. And we can see here that the Porites-dominated reefs have actually been doing pretty well, especially since the 2019 bleaching event compared to those other coral communities. And this is notable, but I want to take a moment and stress that this doesn't mean that low coral cover or degraded reefs are preferable to higher coral cover or more pristine reefs. These low coral cover reefs may still lack certain ecosystem services that higher coral cover reefs are able to provide. But our findings do suggest a different view of resilience, one where chronic stress and human impact remove sensitive individuals from a population and leave behind a system with lower diversity, but more stability due to the dominance of hardier individuals. And these degraded reefs can still benefit from conservation that focuses on reducing local human impacts, and their conservation shouldn't be overlooked in terms of this spatial prioritization. A final possibility is that these 3 different reef types that we identified represent different stages of reef development, where progression through these stages is dependent on the frequency and severity of disturbance. On coral reefs exposed to high wave energy or chronic degradation, you have weedy or stress-tolerant corals that are able to gain a foothold without much competition from other corals. But as disturbance becomes less frequent, these more sensitive but fast-growing corals are able to gain more of a foothold, eventually outcompeting those stress-tolerant corals, especially on incredibly sheltered reefs, where you can get this high coral cover but low diversity community that's called a climax community. This process is called succession, and it plays out over many years on a coral reef, and it can be repeatedly interrupted by disturbance events. So to illustrate the successional dynamics of our sites, I'm going to walk you through a type of plot called an ordination. An ordination allows us to visualize how similar different benthic communities are to each other based on the abundance of different species. And this different species of coral and algae that we measured is called multivariate data. It basically means that we measured more than just 2 variables. For this plot, I don't want you to worry about the axes. All you need to know is that the closer 2 things are to each other on this plot, the more similar they are. And each point here represents a different site. So for example, this site from Kaho'olawe is more similar to this site from Molokai, and they're both pretty different from that site from Lanai. So we can layer on these words represent those different species and reef attributes that we measured. That's our multivariate data. We can also put on these ovals that show the rough position in the multidimensional space of our different community types. So you can see how reef type 3 is associated with Montipora, and then reef type 2 is associated with branching Porites compressa, CCA, and more rugosity. And then you can see how reef type 1 is associated with more Porites lobata and turf algae. So simple enough, right? Easy to interpret. Now let's add in the rest of our data. So here you can see data from all of our different Maui Nui sites shown for 2017, 2019, 2021, and 2023. And we can see some of the same spatial patterns we saw on our map earlier. We can see how those Maui sites tend to be in that reef type 1 and reef type 2, and the Molokai and Lanai sites tend to be in reef types 2 and 3. And so it's not really immediately clear from this figure how those sites are changing over time. So what we can do is we can connect the surveys from each individual year with an arrow. to show how these communities are moving through multidimensional space over time. And so once we do that, we can see that almost all of those high coral cover Montipora reefs are moving in unison towards becoming that intermediate type 2 reef. And this could be an impact of those multiple disturbance events shifting those climax communities back towards an earlier successional stage. If we shift our focus to reef type 2, we can see a lot more stability. Those reefs tend to be sort of circling around the same point in time. So maybe that diversity is a stabilizing mechanism. And then in contrast, if we look at reef type 1, we can see more random changes through time, including this one site, which is actually from that video that I showed you earlier with the red and blue, that the site with the wave damage. So you can see how on a site exposed to more frequent disturbance, a big disturbance can really set you back to an earlier successional stage. But even with this large area times or data going back to 2017, it's hard to talk about successional dynamics because that can play out over decades. So we can do that partially by pairing our time series with longer term monitoring data, not using the 3D models, but traditional transects that was collected by the state of Hawaii with their coral reef assessment and monitoring program. And this is for 3 of our resilience assessment sites. And you can see remarkable stability over the past 2 decades. If I had jumped in the water as a 6-year-old, the reefs would have looked pretty similar to how they do today, which is pretty remarkable. So while reefs do indeed face threats from human impacts at both global and local scales, it's way too early to throw in the towel. These are resilient ecosystems that can persist if we give them a fighting chance. So I've thrown a lot at you today, but I want to bring us back to our resilience assessment. Our predictions did not align with actual resistance and recovery. So where does that leave us? Based on what I've shared today, I wouldn't recommend using resilience assessments as a form of conservation triage, at least not in isolation. If we're going to use those predictions to prioritize locations for protection, we need to validate those with long-term monitoring data. Instead, I suggest that we rely more on feedback from local communities to identify which reefs they feel are conservation priorities. Then we can use large area imagery or some other form of visuals to measure functional indicators and create a scorecard of reef condition, but also to document visually how that reef is changing and to build support for conservation and communicate what types of conservation are needed in that specific area. So I want to share how large area imagery visuals can be combined with animation to communicate environmental change and hopefully impact conservation. And this is an interactive map of our monitoring program that my sister Zoe actually made. She's a professional animator, and we've been working together since I moved to Philly to build this interface and share highlights from my PhD research. And our plan is to incorporate our 3D models, graphs from my thesis, photos from our fieldwork, and environmental data layers to tell a unique story of change over time for each site. The goal here is for the general public to be able to navigate around and explore what their local reefs look like and how they're changing over time. And by hosting this interactive at a public-facing aquarium like the Maui Ocean Center, it could serve as a mechanism for increasing awareness and support for specific conservation measures in specific places. So with that, I'd like to just take a moment and reiterate the key points from the talk today. First, coral reef decline is not a foregone conclusion. Reefs are naturally resilient and they can persist with our help. We need global action to reduce climate impacts, but also local site-specific conservation that's tailored to address the unique circumstances of each reef. Studying reefs at the correct scale is crucial if we're going to understand and predict ecological change, and multi-scale data can help us design these more effective conservation measures. Luckily, conservation technology, like large area imaging, can help us study reefs at multiple scales, and we must ensure that there's equitable access to this form of conservation technology. We should use this technology to better develop an understanding of how resistance and recovery relate to resilience and what indicators are actually driving those things. And finally, I want to make a plea for us to not ignore the degraded reefs. As we've seen today, they have the capacity for resilience and should not be overlooked in the context of conservation triage. So with that, I'd like to go into some thank yous. I guess before I launch into any specific people, I just want to thank all of you here, both in person and on Zoom. As you go through a PhD, it's a very long process, and in any single day or week, it doesn't feel like you've all of a sudden created a huge community, but it sort of builds over time. And then a day like today, you get to see the impact of that. And I'm just grateful to have all of you here. From a research perspective, I want to thank my committee, especially Jen, for just really placing so much trust in me to pursue the questions that I find interesting, to take an existing project and add my flair to it. And it's that freedom to be able to go off to those cool places that you had on the map before has just been really great, and I've really appreciated that. To my other committee members, Lisa, I really appreciated going to COP with you. That was so much fun to see how you interacted with those policymakers. Stuart, brainstorming about ecology or stats or whatever fun topic we were talking about is always such a good intellectual exercise. Eric, thank you so much for being open to evaluating the Nature Conservancy's resilience assessment and being transparent about how this science is conducted and how we can make it better. And then both Eric and Kate, thank you for helping to increase the policy relevance of my research. And I'm really happy with how it ended up, thanks to your help and guidance. Jen was mentioning earlier that the Smith and Sandin lab families are very large, and I've been so fortunate to be a part of that large lab family. I couldn't fit everyone on this slide because there's so many people in that lab family, but I do want to mention a few specific people. Dr. Jill Harris, who was one of Jen's early PhD students, who I actually worked directly with at the World Wildlife Fund. She helped to show me that this type of coral reef research was possible, helped to point me in the direction of Scripps and to show that this was a possible career path. And I mentioned Emily Kelly earlier and how transformative she was in getting this program started. It's funny, we've worked a lot over the years together on Zoom since she left the lab before I started. And the only time we actually met in person was at COP by chance in Egypt. And we just played hooky and went snorkeling together in the Red Sea. And we're like as far away from Hawaii as we possibly could be, but are like two Hawaii people from Scripps. And then we bumped into Christian afterwards, who was, so it was just so weird, such a weird day. And that was a lot of fun. I also want to thank other people in the lab who have helped to train me. Sam for taking me into the field and getting me all set on all the Maui stuff. Adi for creating such a welcoming lab environment and for creating all the amazing coral artwork that's in this presentation. And to the grad students who have come up sort of after me. And Ami, it was great to go to Micronesia with you and to explore the depths in the sub. And to Mitch and Sarah for going to Maui with me. It's been so great to watch you guys grow as researchers and scientists. I want to thank my undergrad research volunteers who made this research possible. And then also our Maui family who has hosted us. I mentioned them earlier in the presentation, but they allowed us to stay on their couches and in their houses. They took us out on the water way below the market rate. They helped us to interpret our findings when we weren't entirely sure what we were seeing. And they're just such generous people. And I really, truly hope for a speedy recovery for that whole community. For the Scripps community, all of you guys here today, I want to thank you guys, especially it really does take a village to raise a PhD student. The dive office for getting us trained up and so I could actually go do my research. This research really wouldn't be possible without you guys. The 2019 dive class was such a educational experience for me, and just being able to regularly dive in the field and at Birch, fulfilling that childhood dream of getting to dive in an aquarium and inspire the kids and feed a fish and all that stuff was so much fun. To CNBC and the grad office and facilities just for helping all of Scripps run and making it easier to be a grad student. It's not always easy to be a grad student, but you guys make it easier. To the comms office, especially Rob Monroe for helping to elevate our work and especially for sending me to COP. It was, as I've been mentioning, a really great experience to see how climate policy is made and how our research relates to that. And then to just the general student body, Queer at Scripps, the 2018 grad student cohort, and just all my friends at Scripps. You're great colleagues, but importantly, you're even better friends. And so thank you all for your friendship. To my funding sources, but also to other collaborators outside of Scripps, I got to do some pretty cool collaborative research going to the Quest Field School on the Big Island to get more acquainted with dive sampling techniques after doing the scientific dive course here, to doing research with SJSU in Maui and helping them with their research, to going on the Nat Geo Pristine Seas cruise or going to Palmyra. It's just all been really incredible opportunities, and everyone's just such a good collaborator. And also to past mentors and teachers and colleagues who have helped me get to the point of even going to grad school, particularly folks at Meridian Institute who read over the GRFP application and really pushed me to pursue this as a career opportunity. I'm just really grateful to all of you. And then To the Hub's homies, I'm confident that my time at Scripps would not have been the same without you guys. You guys were there every step of the way, literally everything we did, whether it was just like me eating breakfast in the beach house to us going to get ramen or going to taco stand or whatever we were doing. And you guys have been such a solid community that it felt more like family rather than friends. And I'm just very, very appreciative of that. Then to my actual family, the very large community of people and my college friends, the Condor Alliance, that just such a big group of people on the East Coast that it made it hard to be here, but in a good way. It made me miss home. And whenever I would go home and come back, I would come back feeling very rejuvenated and supported to go off and pursue all the stuff that I've done here. And I'm very grateful for everyone from that community that was able to come out and visit while I was at Scripps. It just really it just brightened my day whenever you guys were able to come out here. And I really appreciate that. And then finally, some of these pictures have been up here already, but I just want to thank my parents and my sister Zoe and my dog Milo for helping to make this journey possible. They just, whether it was encouraging my passion or telling me to be more open-minded or willing to accept change, they helped to push me and encourage me and get me to where I am today. So I'm appreciative of that. And then to Dr. Chance Dunbar, he beat me to it by like 6 months faster. He's been a reliable partner, a constant cheerleader, a source of entertainment, as I'm sure any of you who know him can attest to, my literal dive buddy and a steadfast friend. And I'm just excited for where the rest of the journey takes us. And with that, I will take any questions. On Zoom or in person, questions are welcome. Yeah.
Great conference. That was fantastic. You mentioned in your talk that resilience, recovery and resistance can be differently. Were you able to tease apart what causes them to react differently? Like what's driving recovery versus what's facilitating resistance?
Yeah, it's a really good question. So for people on Zoom asking if we were able to tease apart what's driving resistance and recovery on different reefs, it was a little bit tricky with our design of our study because we were only looking at 6 sites. So we weren't able to have like a statistically steadfast rule on that, but just looking at the individual site level, it appeared that it probably interacts a lot with the local environmental conditions. For example, at Kahakili, you have that's a site that was near the wastewater treatment plant where there's been a lot of herbivore protections. And so it's possible that it would be great if we could have had herbivore data in there, because just having more herbivores could have allowed some of these corals to be able to better resist stress. And especially that would probably be more on the recovery side, better to control those algae blooms and to help corals not get overgrown after a bleaching event or something like that. But it really does seem to be, at least with our large area imagery data and our time series, very site specific, which can be challenging and frustrating from trying to come up with a general rule. But because these models give us such insight into what's happening, and if we pair that with other knowledge about the environmental conditions in that location, we can try to come up with these very specific interventions to address, like at Keavakapu, what might be driving that decline of those specific corals, and just try to use that as sort of a detective work to figure out a solution for each site. Great question. Olivia.
So you often hear that like the animals or coral reefs or whatever is not going to be able to adapt best enough. Now, I was just wondering if you saw anything in your data that like resilience rate or the hesitancy is
Yeah, that's a great question. So the question for Zoom was if we're seeing, because climate change is getting more intense, if we're seeing the reefs not be able to keep up with increasing disturbances from climate change. And so I mean, up before the 2015 bleaching event, there actually hadn't been a lot of major bleaching events or other climate-related impacts on Hawaii's reefs. I know there was a hurricane back in, I think, the 90s, but there definitely has been more frequent disturbances in the intervening years. And we haven't necessarily seen a protracted decline at most of our sites. Some of them, as you saw today, but some of them they sort of stabilized or still were able to recover. So it seems like at least not yet. It hasn't gotten to the point where the sites are being hit and then sort of stepwise declining. I mean, that always could happen. And it's sort of this model of resilience. Maybe that will, the corals that are left after each disturbance will just be truly the ones that are able to persist, like the type of corals you might find in a really highly impacted harbor or something like that, just really hardy individuals. Or it could be it surpasses a threshold or even they are not able to surpass that. And it's hard to say just based on this data alone, but good question. Bev.
You had this focus on conservation tech, and you have a lot of thinking and a lot of conversation about how conservation tech and what the barriers to adoption might be, as well as kind of what what the most promising tools might be. So I'm just curious, from synthesizing all of that, what is your perspective on what some of the most promising tools for conservation tech are, and what are some ways that we can use more?
Yeah, that's a great question. I mean, in terms of, so I guess for folks on Zoom, what are some other promising examples of conservation tech, and how can we just make conservation in general, this tech, more accessible for conservation? And so I know for large area imaging, one of the insights from our survey was that people were facing different barriers based on where they were. Like if you're a new user, you're going to face very different barriers than if you've already started using it and then you start running into roadblocks along the way. So just trying to get a sense for whatever conservation tech it is, like what are sequentially the barriers that people are going to face, and trying to set them up from the beginning to be able to better plan for what they're going to need to navigate as they're trying to address those barriers. Because I mean, money is almost always a perennial barrier, but even if you look more in depth throughout the process, it might be, in this case, access to cameras at the beginning, and then later having the people to actually collect the data from the 3D models. And then you get to like our stage where you have so much data. How do you store it? How do you serve it to people? And so I'm sure that there's probably allegories for environmental DNA or other types of remote sensing. And as we push the boundaries of the science to try to get to a higher and higher resolution, other people might be still struggling to onboard the most basic elements of that technology. And so just as researchers pushing the frontier, not forgetting the partners who are a few steps behind us and helping them to come to where we are so that everyone can be using this transformative technology. Great question.
One follow up on obviously the photogrammetry is top notch. If you like something the performance of the face, what sorts of where was the TNC data coming from? With GIS or like survey based? And then is there any way that you imagine that it's in a world where maybe it's not going to be possible for everyone to access photogrammetry, so like kind of model a connection between the two data formats, so that if you could still be collecting this like economically feasible data, but then like making it more accurate based on the model that applies photogrammetry to the more basic stuff.
Great question. So for people on Zoom asking about specifically what the Nature Conservancy was using to collect their data, but also if there's ways to bridge the gap between people who may never be able to use this technology and people who are, and if there's ways to interpolate that and sort of connect the dots, if I interpret it correctly. So I think for the nature, to answer the first question, the Nature Conservancy was doing sort of traditional transects where they were counting corals. They also had photoquadrats that they were using to measure coral cover and look for coral recruits and stuff like that. So it was low tech in the sense that it's still using pictures, but not creating a 3D model. So a lot easier computationally and faster in a lot of ways. In terms of other ways of sort of for people who can't have access to this technology, I mean, in some cases that can be like people, this technology could be not a good thing. I remember talking to one survey participant who worked in the Philippines and had local divers that were going out and surveying the reef. And the people who were actually analyzing the data were based off-site, and those local divers that were collecting the data and able to communicate that story to the local community were actually, through that, building support for protecting that local reef. And so implementing conservation isn't, or technology to help conservation isn't always going to work in every specific case. You need to make sure you're not undermining a system that's already working. And so there's plenty of merits to the low-tech approaches as well. It's really, if you want to be studying multiple scales, change over time, that sort of thing, where it becomes super handy. And I think that there's been a lot of conversations and efforts about, can we have some sort of learning network or institution that is serving as a knowledge hub for whether it's this technology or some other form of conservation technology to help build capacity in sort of fledgling users, or to help connect people that might be facing the same problem. And so I guess that's less on the data interpolation side, but more on the connecting people and trying to solve problems that way side.
That supplements the visual evaluation of the technology that you're talking on, that takes samples and evaluating what's going on internally with it.
Yeah, that's a great question. So for people on Zoom, the question was, are we doing something besides just sort of the visualization and looking at the model to get our data? And that's part of why the PhD took so long. It was like each time we have our 10 by 10 meter site, we're analyzing to get percent cover. We look at 2,500 points on the reef and identify what was on the reef under those points. So for all these, probably hundreds of thousands of points that I clicked through to look at the reef. So sometimes would close my eyes at night and still see corals, and would curse them for still being in my vision when I was trying to sleep. So yes, it takes a while to collect that sort of data. We can get structural complexity data. We're actually measuring the surface of the reef. For the bleaching stuff, we were actually tracing the outlines of individual coral colonies and using that to quantify if they were growing or shrinking. So lots of ways to get that data directly from the models. Right now, that takes time, but there's increasingly pipelines to reduce the amount of time and to make that more automated. We're still using like a human in the loop system so people can check to make sure it's accurate, but I'm sure in the future it'll keep getting faster and faster, but we're also going to want to measure bigger and bigger spaces. So there's like this trade-off between how long it takes to collect data and how big of an area you can measure.
First of all, congratulations. This was a tremendous presentation. So you talked about the different reef types and the different levels of resilience that they have and how they can transition from one to another. Do you have any reason to think that the more traditionally resilient reef types that have more diversity might eventually step down into a more resilient, more actually resilient reef type?
Yeah, it's a great question. So this question was focused on the succession and jumping between different reef types, and it was, I think the question is, can the more high coral cover reefs eventually drop down to the lower ones? And I think that's probably what happened over a long period of time on Maui, because before people, there aren't really many oceanographic differences between those islands. They're all pretty close. And so before that sort of stepwise process, those reefs probably look pretty similar to the other islands. And so I think that sort of could play out in other places if you have similar amounts of human impact, or maybe the climate impacts over time. So great. I'm going to look at Zoom and see if there's any. We have one question about, were you able to use CoralNet to automate any of this process? And CoralNet is a UCSD made tool that allows you to automate any of the images. I didn't, but there's some other people in our lab that are using CoralNet, and I was using the 3D interface. You have access to multiple different images for any point and can use that to make an informed guess about what the reef is at that point. So CoralNet hasn't quite yet been sort of merged into that pipeline, but I know that there's work being done to do that. So it's hopefully that'll make the next PhD who comes to do this take less time, and they can look at a bigger area or expand this in some other way. Okay, one more question.
Pretty early on, we talked about implementing metrics, and you kind of excluded herbivores early on. Their motion being hard to capture with the cameras and the sensors and the visual spectrum there. Do you know if there's any advancements in ways to better gather that data?
Yeah, that's a good question. So the question was about herbivores and if there's any ways we could have better quantified that. One way that I've been thinking about is using the 3D models to look for little bite scars on the reef where the fish are going around and actually biting the algae. And we actually had a master's student who worked on that several years ago with parrotfish and seeing bite scars on the reef. The one downside is that also takes a lot of time to go look for those. And so if I wanted to stay several more years, I could have incorporated that, but I was ready to wrap it up. So yeah, the next PhD student can do that. Awesome.
Well, I think we might end questions there, at least formally. I would like to thank Orion's committee and invite committee members and Orion to the conference room next door, and invite all of you to stick around and hopefully celebrate some letters.
Yeah, that'll be in
Or
this room, just
behind.
for anyone who's
Yeah.
who's here. Yeah.
Celebrate some new letters added to Orion's name, hopefully in just a few minutes. So thank you, Orion. Great job.
Thank you, guys.
Congratulations.
Thank you, everyone, for joining on Zoom.
Great job, Rye.
Love you.
I don't,
Love you,
I'm
yeah.
the one hosting the Zoom, so it's.
Congratulations. Woohoo. Woohoo. Woohoo.
Awesome job, man.
Yeah, yeah.
Great job, Orion.
Great
job, great job.
Yeah, yeah, Papa.
Bravo, bravo.
Axios, axios.
So impressive.
Good.
Congratulations, Orion. What a privilege and pleasure to be able to watch all this with you.
Yes. That's so true.